Okay, so we keep this whole series as anatomy of questions, and here we want to discuss about the detailed structure of a question, because if you understand the structure of a question very clearly, you can answer them fast. Okay, and sometimes understanding the structure is more important, even important than knowledge of the subject itself. So today we will talk about unit 1 and unit 2 questions. Okay, so 6 questions from each unit. Approximate number of questions you can expect from these units in parts in CSIR NET exam. So this is okay. So we are back with another anatomy of a question session, and this is about unit 12, that is applied biology. So most of the questions from unit 12, you cannot distinguish them separately from unit 13 questions. In fact, many times the questions are combined with unit 13 altogether, like applied biology, applications of biological techniques, combined questions. But particularly unit 12 questions are mostly direct type, memory based, many of them. So you need to know a lot of information to answer these unit 12 questions compared to unit 13, because unit 13 questions are mostly analytical, but 12 is mostly memory based. So let's start to solve few of the questions like the part C question of CSIR NET for this chapter. So let's begin. The following are certain statements regarding the use of agrobacterium in plant transformation. This part is very important for this unit 12. They ask many questions from this part every time. So this question is from agrobacterium mediated gene delivery in plants. So if you know that, you can answer this question fast, otherwise leave it. There are options. The statements are given. Agrobacterium tumefaciens causes crown gall disease, and A. rhizogens causes hairy root disease. This is one of the basic thing that you know when you prepare this chapter. And we know, yes, it is true. The statement 1 is a true statement. Statement A is a true statement. Now, if you look at the option now, without reading the rest of the statements, that's the study. That's how you understand the anatomy of a question. If you look at the option, the only 2 options are present with statement A. So our answer is somewhere present between option 1 or 3. So we can easily eliminate option 2 and 4. 50% chance now. Now statement B, region A in TI plasmid is responsible for replication. And statement C, region D in the TI plasmid is responsible for virulence. Now you need to know the structure of the TI plasmid, and if you recall the structure of TI plasmid, we always write like vir D genes, and there are different types of vir D, 1, 2, 3, 4, 5, 6, 7, a different vir D. So D obviously gave rise to the virulence, and that is the region D. So if we know that the region D is associated with the virulence impact, then we can select that option 3 is the correct one and 1 is wrong. If we read the oncogenic region in the tDNA is responsible for the unusual amino acid synthesis, the answer is also wrong. This statement is not correct there. So because the oncogenic portion of the DNA that helps in producing auxin, that helps the growth of the plant cells, that's how the tumor is formed, the crown gall is formed. So we know that A and C are correct, so we select option 3 in this case. So this question depends on your understanding of the TI plasmid, whether you recall the structure of the TI plasmid or not. Okay, so this is the overall big picture of how exactly agrobacterium mediated gene delivery is done. If you look at the TI plasmid, this is the vir D section, and apart from that, this D region, there are also vir D, vir B, vir F, and many other vir complexes as well present there. Okay, so you need to remember, because for the responsible for replication, that is the ORI side that is present separately. Okay, so if you know this process, particularly this picture, you can answer these questions. Let's move to the second type. Marker assisted selection or MAS, defined as selection based on the molecular markers, should have some important criteria for the plant breeding activities. Some statements about this criteria are mentioned below. So this question is from marker assisted selection or MAS. If you don't know about marker assisted selection, you cannot answer this question. If you heard this term for the first time, you cannot answer the question. If you know, then only attain it, otherwise leave it. Now statements are: markers should co-segregate with the desired trait of interest. B, markers should not co-segregate with the desired trait of interest. Markers should not unlinked with the desired trait of interest, And fourth one, marker is used for indirect selection of genetic determinant or determinants of an interest. Now you need to know at least one to start with. Among these four statements, the easiest one is the D1, the statement number D. Marker is used for indirect selection of a genetic determinant or determinants of a trait. We know markers are not a direct selection parameters, because markers are molecular markers that we use for marker assisted selection. So we cannot just select based on the height of the plant or color of the flower, no, but we are looking at a gene, the presence of a sequence, which is an indirect marker of the phenotype. So marker always used as an indirect selection. Okay, so if D is correct, we know option 1 and 2 are wrong, so either option 3 is correct or option 4 is correct. Now we need to know whether the statement A or C, which one is correct. Now among them, marker should be unlinked with the desired trait of interest. This is definitely not true, because if the marker is unlinked from the desired trait of interest, then why we are using it as a marker? We are using it as a marker because it's definitely linked with the desired trait of interest. So we know C is wrong, so option 3 is wrong. We only left up with option 4, so option 4 is the correct option 100% with 100% surety. But if you look at the A statement, marker should co-segregate with the desired trait of interest. Yes, this must be true. If you don't know this statement A from the beginning, but still knowing statement D and knowing that C is wrong, you can still answer this question quite fairly. Now let's look at this question. The following genes have been genetically engineered to develop herbicide resistance in plants. Statements are provided: resistance to glyphosate using 5-enoyl pyruvyl shikimate 3-phosphate synthase gene. Second one is bialafos resistance using the bar gene. Sulfonyl urea resistance using the acetolactate synthase gene, and atrazine resistance using the glutathione-ase transferase gene. So a lot of information, and these are hardcore plant biology questions. So if you're not from botany background, you probably haven't prepared that, because we haven't talked about that in the class. We don't go for it. Only the biology, like agricultural students, can attain this and can be can answer this question, because it's also applications of botany as well, applications in agriculture as well. So what we should do here is that you need to remember, you need to know the function of different herbicides with the different genes associated. Otherwise you cannot answer that. So actually I'll give you a table that explains everything. You can see easily how there is glyphosate as well as the sulfonyl urease, how they are related to acetolactate synthase, or the 5-enoyl pyruvyl shikimate 3-phosphate synthase gene, and how they are linked. So we know glyphosate is with this one, and and this acetolactate is by sulfonyl urease. So based on these statements, we can select option 4, that A is correct and C is correct. Both are marked in here. So this is a table, we are not familiar with that. Even particularly, if you're not from this background, you cannot attain it. You should not answer this question. But this is a 100% memory based question. You cannot answer that without any way. That's why I don't like this part 12 questions mostly. Somatic embryogenesis is an important exercise in micropropagation and genetic engineering of the plants. The following steps are considered as critical for achieving somatic embryogenesis. So there are statements regarding somatic embryogenesis. We have talked about somatic embryogenesis, so we will attain this question. Reducing the concentration of sucrose in the medium by half. Addition of hormone 2,4-dichlorophenoxyacetic acid 2,4-ADD. Induce the somatic embryos. Reduce sugar concentration to 0.6% weight per volume. Reduce maltose. Use maltose in place of sucrose as a carbon source. Now you need to know few important steps regarding the somatic embryo formation. And few of these things, the first thing is that we need to induce the cells to form the embryo, because normally if you take a cell from the plant and if you put it into the culture media, the cell will grow into an adult plant. They will not form any embryo. It's like the totipotent nature of the plant cells allow them to grow into an adult plant. So what makes the difference? By converting a plant cell into a somatic embryo rather than forming an adult plant. That's important about this. And we know one thing is that for that reason, those cells need to be induced by the presence of a hormone 2,4-dichlorophenolic acid. If you don't know that, you should remember B is correct. If B is correct, then we can eliminate option 1 and 4. Now we also know that sucrose is not generally used in this case as a primary sugar source. If we use sucrose, then the plant will take it up quite easily and grow into an adult plant. So we need to use maltose instead of sucrose. So D is also correct. That means option 2 carries both the information, is a correct one. So knowing these two important features of the somatic embryo, we can answer this question. So the production of the somatic embryo, we take out the plant cell, put it into the culture media, we supply them with 2,4-dichlorophenolic acid, okay, as well as, if I write it down here, 2,4-DAA is required for it, reducing sugar consent, reducing any of this. If you look at here, the concentration of sucrose, reducing concentration of sucrose in the agar, it will not have any impact. Maltose is used in place of sucrose as a carbon source that can influence the production. So these are the steps of how the cell taken from the plant can form into somatic embryo, and how it continues with the changes of polyamines and auxins and the concentration of that with changing parameters. Okay. Now let's solve this one. Factor 9 is essential for blood clotting. Deficiency in factor 9 could be corrected by delivering factor 9 gene using viral vectors. In an experiment, the gene for factor 9 was cloned appropriately into adenovirus, adeno-associated virus and retrovirus. The retrovirus integrate the gene into the dividing cell, full integration of the gene. Adeno-associated virus integrates partially into the non-dividing cells, okay, and AV, adenovirus, does not integrate the gene but transfects both the dividing and non-dividing cells. Following expression profiles for factor 9 was observed when the 3 vectors were injected intramuscularly into 3 groups of experimental mice. Which one of the following outcome is correct? So this question, you can either take it from unit 12 or unit 13, doesn't matter. This kind of question is generally from the gene therapy idea, that sometimes to correct the deficiency of a gene, we need to transfer the good copy of the gene from outside using a vector, a viral vector. And we know depending upon the type of vector, the assembly and integration of the gene differs. So in this case, we know the retroviral vector integrates 100% proper integration in the dividing cell. So this will spread. For the adeno-associated virus, partial integration, so little less, and adenovirus, no integration. So if you look at the graph, it's showing the, after the process of gene delivery is done, we are looking at the time in days in x-axis, and we are looking factor 9 present in the serum in the y-axis. So obviously, if our target gene is integrated, then we will see a spike in the production of factor 9, and the maximum spike we find out is here for B, okay, and it is sustained for maximum duration. That means B is done by retroviral vector. A, you can see a rapid spike, but then it's a fall. So definitely it's from adenovirus, although the transfection is done, but there is no integration, so there is a gradual drop. Okay, and for C, the partial integration, adeno-associated viral integration. So you can easily explain this graph if you know how they are going to integrate, which is also told in this question itself. This is the beauty of this kind of questions. If you read the question couple of times, they already have shown you the answer. You just need to look into the graph, and if you can interpret a graph data, you can easily answer this question. So which option here carries all this? The option number 4, A by AV, B by RV and C by AVV. So option number 4 must be the correct one in this case. Okay. Now let's look at this one. A protein undergoes post-translational modifications. In an experiment to identify the nature of modifications, the following experimental results were obtained. A, protein moved more slowly in a SDS page. B, isoelectric focusing showed that there is no change in the pi value of the protein. See, mass spectrometric analysis showed that the modification was on serine residue. So these three statements, these are a fact regarding the protein. So they are not like some statement are true, false. So these are all true statements. So there is some sort of post-translational modification is done. Afterwards, we find out no change in the pi and the modification is done in the serine. We know that, and we also know that after this modification, this protein is moving slowly in the SDS. So that means the protein is becoming little bulkier after the modification. From this statement, we get that it's bulkier now. So modification to serine means, obviously among the options, if you see phosphorylation, glycosylation, ubiquitinization and ADP ribosylation, among these four, the one modification that is done in the serine residue most is the glycosylation. Okay, and due to the glycosylation, we also know the protein become bulkier, and as a result it will slow down the movement in the SDS page. So obviously the option 2 is the correct one. So among these four, you can easily eliminate the phosphorylation, you can eliminate the ubiquitinization because they are not even involved with it, and ADP ribosylation, if there is any change in that, then also you can see there may be a chance of the change in the pi. So we know that glycosylation, and also it does not take place in the serine residues. The change in the serine residue means it's either like SH and OH bonds, so disulfide linkages, or in this case glycosylation. So this is the proof that the modification is done in serine residue can help us to go very close to answer this question. Although there is no combination provided here, but still you can answer the question knowing that modification is in the serine, and as due to the bulkier modification, you will see that in the SDS page the movement of the protein will become very much slow. Okay. The following statements are related to plant tissue culture. Okay, so another question from tissue culture. Friable callus provides the inoculum to form a cell suspension culture. The process known as habituation refers to the property of a callus losing the requirement of auxin and or cytokinin during the long-term culture. This is true, we know that. And even callus cellulose and pectinase enzymes are usually used for generating protoplast cultures. During somatic embryo development, torpedo stage embryo is formed before heart stage embryo. So this question is from your overall understanding of the tissue culture. If you know tissue culture, the basics, you can answer this question. First of all, you need to know at least 2 to 3 different properties to answer that. The process known as habituation refers to the property of callus losing the requirement of auxin to cytokine ratio for their growth. This is true. We know that the B is true. If B is true, then we know option 3 is definitely not true. Now let's read more. Cellulase and pectinase. Pectinase and cellulase, these are the structures, the integral structures to hold the cell outer structure, that is the cell wall structures. So if we use this cellulase and pectinase enzyme that can break down the cellulose and pectin, which forms the outer structure of the cell wall, usually can destroy the cell wall that is much needed to produce protoplast, where we don't have any cell wall outside of the plant cell, we only have the membrane. So obviously we need that. C is correct. Once we know that the C is correct, then obviously what we know is that 2 is wrong, option 2 is not correct. So now we are 50-50 chance, either 1 or 4. Now let's read the last one. During somatic embryo development, torpedo stage embryo is formed before heart stage. This is wrong, because torpedo stage is the last stage. If you look at here, this is how it works: the globular stage, heart stage, torpedo stage and cotyledonary stage. I mean, torpedo is the like last to last stage there. So these are the sequential stages. It is told that the heart stage came after torpedo, but no, torpedo came after the heart stage. So this D statement is wrong, and if D statement is wrong, then option 4 is wrong. So we are left up with option number 1. But this question needs you to know at least 3 out of 4 statement to answer. Generally most of the statements, most of this kind of statement combination questions need you to know 2 out of 4 question to answer, but this one required 3 out of 4 to answer this correctly. Okay. Fluorescence recovery after photo bleaching in live cells is used to determine which of the following. You can't get this kind of question most of the time, because this can be solved within 2 to 3 seconds if you know the process. And this is a part C, meaning group C question. Group C question must be big and tough, but this one, it's a direct question. Now, fluorescence recovery after photo bleaching is used for what? Co-localization of proteins, distance between two organelles, definitely not. Distance between two organelles. Diffusion of proteins, yes. Nucleic acid compactness, definitely no. Co-localization, definitely no. Co-localization studies is done by the other marking of the proteins with different tags in the vesicle transport system. So this is definitely not the case. Fluorescence recovery after photo bleaching, known as FRAP, is actually done in the plasma membrane to check the movement of the membrane proteins from one place to the other, diffusion of the membrane proteins from one place to the other. Where you have unleveled cell surface, then the cell surface is leveled with fluorescence dye, then we put the laser beam that can take out the bleaching of the area. That means it will bleach the area, few region where the fluorescence dye is out of the action, and then we wait for some time when the when all the proteins and the phospholipids start moving towards the bleached area, and slowly we will see in the bleached area, when after the bleaching the intensity of fluorescence dropped, but now slowly we will see the intensity to rise gradually. That means the proteins as well as the the proteins which are tagged with the fluorescence dye is moving towards this bleached area. So this is going to prove us the diffusion of proteins in the cell membrane, right? So if you know this, you can answer that within blink of a second. So this kind of question you can get in unit 12. Yes, the answer is yes. If you ask yourself this question, that can you qualify CSIR NET Life Science exam within 6-month preparation? Yes. And if you don't need to believe me, just see the results that our students are getting who joined Shomu's biology coaching for 6-month period of time.
So I'm really thankful for Shomu's biology, and I would suggest everyone to go for it.
A lot of help and support by Suman sir always remembered in my mind.
The study materials I got helped me to understanding the topic very well, and the test series helped me to covering up the syllabus in a perfect time.
This is all because of the online test series.
In-depth knowledge what Shomu's biology gave me. I want to thank Shomu biology. I want to thank each and every member of Shomu's biology for their effort, whatever they are putting on us.
And I am very much happy to choose Shomu sir for my CSIR NET JRF journey.
It really leveled up my preparation. I was confident on the day of exam.
The classes helped me a lot to understand my subject from very basics to advanced level.
So I really thanks Shomu sir and also ma'am for always guiding me.
With proper guidance, roadmap, and the help and training with perfect amount of study material and how to utilize them, the resources, everything together can help you qualify that. I am the person who will help you reach to the pinnacle of your success and your journey for the CSIR NET Life Science examination. If you trust me, if you trust my lectures, then join Shomu's Biology online coaching. Registration is going on. The details are there in the description. Fill the form to reach out to us or simply contact the number given in this video.